Welcome to Animated Science Biology. We are continuing aerobic respiration, moving into step 2 and step 3, which is pyruvate oxidation, also called pyruvate decarboxylation, and the citric acid cycle, which is also called the Krebs cycle, and is also called the TCA cycle. So there's many names for it. Whatever your class calls it, just remember that. It doesn't really matter. I learned the Krebs cycle. It's gonna be the citric acid cycle from now on because everybody wants to get rid of the names of scientists that put their name on a certain process. Anyways, glycolysis took place in the cytosol. We started with our glucose. So glycolysis is all about splitting that glucose and making energy out of that glucose. Okay, so we started with glucose, 2 ATP and 2 NAD. We took those molecules through 10 steps and 10 enzymes, and we finally created 2 pyruvates, 2 ATP, so 2 net ATP, so a total of 4 ATP, and 2 NADHs. So if you haven't seen that video, go check that out. I will put a card up in the top right, so click that if you haven't seen that. So in these 2 steps, we're going to use our 2 pyruvate molecules. And these 2 pyruvate molecules, they are either going to be funneled into this pathway, which is the aerobic respiration pathway, but we need oxygen for them to get funneled down this pathway. If there is no oxygen present, they're going to go into fermentation, which I will make a video on in the future. But for them to go down the aerobic respiration pathway, there needs to be oxygen. Before we continue on with pyruvate oxidation, we first need to talk a little bit about the mitochondria. And if you've taken any biology class ever, everyone knows the mitochondria is the powerhouse of the cell. Yes, it makes energy, okay? That's what we're talking about. First, we need to talk about the structure. So it's got an outer layer and it has an inner layer. So there is an outer membrane and there's an inner membrane. That's what they're called, outer membrane, inner membrane. There's also 2 compartments. The matrix, which is on the furthest inside of the inner membrane, and the intermembrane space, which is between the membranes. So if we zoom in here, we can see all of those layers and the compartments there. And at some point, pyruvate is going to get transported into the mitochondria using transport proteins. Those are out of the scope of this video, so we're not going to really go into that. But once pyruvate gets into the matrix, it's going to encounter pyruvate dehydrogenase, which is a huge complex enzyme, which this is even a simplified look at it. But there is definitely 3 important subunits. So pyruvate encounters the first subunit. It is a 3-carbon molecule which is split into a 2-carbon acetyl group and a 1-carbon CO2. The acetyl group is attached to a coenzyme A molecule, and CO2 is going to be a waste product. And so we're going to keep that score in the top right here. In the meantime, a hydrogen from the acetyl group is going to reduce the NAD molecule into NADH. So that is all for pyruvate oxidation. We have a CO2 created and we have an NADH created, and we also have this Acetyl CoA created, which is the coenzyme A with the 2-carbon acetyl group attached to it. So for the rest of the process, for citric acid cycle, if we count the carbons, that's going to show us what is occurring. Okay, so we're going to be following the carbon molecules, and coenzyme A is just going to act like a taxi. Okay, we're going to use it again at another step, but just for this step, coenzyme A is taking the acetyl group to the first step of the Krebs cycle. or the citric acid cycle. So Acetyl CoA delivers its acetyl group to this first enzyme. This enzyme is going to remove the acetyl group from Acetyl CoA and attach it to a 4-carbon molecule known as oxaloacetate. Some classes require you to know these first 2 molecules. If you don't need to know it, just know it's the 4-carbon molecule. And that's going to create citrate, which is a 6-carbon molecule. So this 6-carbon molecule, known as citrate, is going to go to the second step of the pathway. And in this second step, we're just going to have a simple rearrangement reaction occur, nothing crazy. We still have a 6-carbon molecule. It's going to go to the third step of the reaction. And this reaction, we're going to remove a carbon unit as CO2. We'll keep track of that in our scoreboard. And during this, we are going to reduce another NADH. And so we'll keep score of that. This 5-carbon molecule is going to go to the 4th step of the pathway, which removes another carbon dioxide and attaches the 4-carbon molecule to the coenzyme A again, so it's going to use it as a taxi. Meanwhile, another NADH is created using a hydrogen from the molecule. The 4-carbon molecule is going to be carried by coenzyme A to the 5th step of the reaction. And during this step, we're going to remove the 4-carbon unit. And we're going to recruit a guanine diphosphate molecule and an inorganic phosphate. And now this is the energy molecule. So we're going to create guanine triphosphate. Sounds very similar to adenosine triphosphate. So this is GTP. So we're going to create a GTP molecule, which later on in the cell we can create ATP from this GTP molecule. So we can take the phosphate from the GTP and add it to ADP to create ATP. And that's a lot of stuff just to say that we created one ATP here. Move into the 6th step of the reaction. We're taking our 4-carbon molecule. And in this step, we're going to have another electron carrier come in, known as FAD. And this electron carrier, again, is just going to carry electrons to the electron transport chain. So we have the reduction of FAD into FADH2. We'll keep that score up in the top right. 4-carbon molecule continues to the 6th step of the reaction. We have a water molecule recruited and rearrangement takes place. Not much occurs here. And then the 8th step of the reaction, we're going to rearrange this structure into oxaloacetate and further reducing one more NAD into NADH, giving us a total of 3 NADHs created. We'll keep that score up there. And then oxaloacetate returns back to the first step to attach to another acetyl group and create citrate again, and the cycle continues. So per pyruvate, we saw 3 NADHs were created, 2 CO2 molecules were created, 1 ATP was created, and 1 FADH2 was created. So since glucose creates 2 pyruvates in glycolysis, and then those 2 pyruvates are both going to go through pyruvate oxidation, we can double these numbers. And so after 2 pyruvates go through this entire cycle, we have 6 NADHs, we have 4 CO2 molecules created, 2 ATP molecules created, and 2 FADH2 molecules created. And you may be thinking to yourself, this is a long pathway, how come I only created 2 energy molecules? Well, most of our energy payoff is going to come from these electron taxis, the NADH and the FADH2. So in the next step, aerobic respiration, the electron transport chain, we are going to see this energy payoff greatly from these electron carriers. So even though we only made 2 ATP per glucose for these 8 reactions here, we will have our energy payoff. Here I'm giving you a simplified model of the Krebs cycle, or of the citric acid cycle. And this is something that you can screenshot, you can download. This will help you study. It omits steps 2 and step 7 because those are just simple rearrangement. And it also eliminates some detail about coenzyme A and the creation of ATP from the GTP. However, this gives you a very good summary for you guys to study, and it shows you where the molecules are coming from. So like I said, if you follow the carbons, it gives you the hint on what to look for the citric acid cycle. So that does it for this video. I hope you liked it. If you did, please give me a like. If you learned something, give me a like. If you have any questions, let me know in the comments. I am pretty good about answering comments, so let me know if you got any questions. Please subscribe. We'll see you in the next video. Electron transport chain will be coming soon. Thanks guys, have a good one.